Hi everybody, it's Mrs. Hunt. This is the 23rd, so it's actually Wednesday, and I'm sorry I'm taking so long to get this video out to you. We've had internet problems and all sorts of issues. So I'm going to go ahead and get us started here. This is the helpful videos for cell membranes, and I highly recommend you watch this. It is part of the Pearson homework assignment, and it really goes through that phospholipid bilayer, and I feel like it does a great job really looking at it. So this week you are studying the plasma membrane and transport in and out of the membrane, and it's really important to understand all the little tiny details about it, but also get the big picture of why this is happening. All right, so definitely watch that video first before you do my notes. All right, I'm going to, and speaking of notes, this is the outline notes that I made for you. So if you're at home, you might want to pull this up and be able to look off of it. I'm going to be teaching over here and going through the notes myself, so you can go back to it. My in-class kids, I made a printed copy because it just seemed to be easier for them. You're welcome to print it out, or if you let me know, I can print one out and leave it for you in the attendance office if you would like that. So just let me know. Okay, I'm going to try to see if this works today. Yay, I think it is working. Okay, I'm going to bring my face over here. All right, so I'm going to be drawing down here, and this is similar to when I used to draw for cellular or for honors biology. So let's start talking about that plasma membrane before we get anything going here. Okay, so let's talk about the plasma membrane. All right, and what do I want to know about the plasma membrane that is made out of those phospholipid bilayer? So when we're talking about phospholipids, we're talking about phosphate here. And the lipid part, underline that right there, that's where we're talking about a fat. So when you guys did your macromolecules, you kind of went through that fat. Bi means two, so this is a two layer that we're having here. So if I take, let's draw an animal cell here. And let's pretend I'm taking a piece of that cell membrane out, okay? So if I pull this out, it looks like circles here, and then these little tails, and then tails, and then a circle. So we can see that this is two layers. Let's say that this part is the outside, okay? And they're going to call this the extracellular fluid. So that watery environment that the cell is in. All right, in here, this is the inside, and this is the cytoplasm side. All right, let's go ahead and remind ourselves that this little guy right here, this is called the head, and this is called the tail. I do recommend that you draw this at home, practice going over it, and draw this with me. So I'm going to go fast, but you can hit pause anytime you want to. All right, the head we call hydrophilic. And the tail we say is hydrophobic. This gets us into our Latin real quick. Hydro means water. P-H-Y-L, phile. You've heard it for pedophile before, which is really bad. Phile means to love. I think in your Greek mythology, Ophelia is the basis of that word, which goddess of love. So phile means love. And then phobe or phobia is fear. So we have water-loving heads, which is good because the extracellular fluid is made out of a lot of water. And then the tails are the hydrophobic. So this is where there's basically no water. And we want to think about it as being like oil, like cooking oil, how it separates vegetable oil from water. This creates a barrier to stop things from moving in and out. Okay. And that's good because that's what we need. The job of the plasma membrane is selective permeability. All right, which means it controls only some things can come in and out. Last week, we talked about some of those things. We said oxygen. We talked about sodium. We talked about salt. We talked about carbon dioxide wanting to come out, water coming in. We talked about the SNOPs, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Those elements need to come in. When we eat amino acids, and those amino acids need to come in to build a protein. So all these things are coming in and out. Glucose is coming in. And so the selective permeability means it controls what comes in and out of that cell. And that's really important. Okay, so really what's happening, these tails and heads, so let me draw over here. The heads really form, actually the tails, form these like connections like this when they're normal. And then the bottom tails are doing the same thing. So we have the tails doing the same thing where they're touching each other. So for example, let's have a little water molecule come through here. Normally, he's okay to pass through the head, but when he reaches the tails, if they don't want him to come in, he gets bounced back out. This area is going to stop movement through the cell membrane. So what the tails have to do is the tails have to change their shape. They have to come in closer. So it's almost like they're next to each other or they're away from each other and they're like, oh, water's coming in. Ooh, get it, let the water come in. And then it comes back to where it was. I'm sorry, my camera is. So this way, the water can move all the way through, okay? And we're going to discover that this isn't the easiest way for water to move through. This is actually pretty slow. And we've got to move a lot of water in that cell. So this takes a little bit of time to be able to move that water through the cell, okay? So there will be helpers in our notes here in just a minute. All right, so I'm going to go back and draw a little bit more. So we're going to see that the phospholipid, there's our phosphates. I don't think I labeled that. Okay, go back over here. The head is actually phosphate, and the tail is the lipid. Okay, so that gives us our phospholipid, and then here is our bilayer. So we have two layers. Okay. All right, so I'm going through making a better model here. Okay. All right. So first of all, let me put, all right, these are carbohydrates, which you remember in your macromolecules, carbohydrates are your sugars. What this guy is he's a signal. and he's going to help in cell communication. So he's hanging outside of the cell, and he's going beep, 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 beep. Okay, oh, this is oxygen, you can come in. Oh, this is a glucose, you got to go over here. So he's hanging outside the cell, kind of like an antenna would be hanging outside of the cell. But he's an integral part of this phospholipid bilayer in the cell membrane because he's signaling where to go. All right, the other thing that is very important in here, I'm going to use a different color, is inside here. All right, this is cholesterol. Okay. All right, cholesterol's job is to loosen up the cell. I think cholesterol is kind of like, well, I mean, it is a fat. So cholesterol is a fat. And what we want is the good cholesterol, okay? This is unsaturated. So fats from like avocados, fish, nuts, olive oil, those kind of good fats. And being a fat, it loosens it up. So in here, it keeps the tails from sticking too close together, and it allows everything to be very fluid inside that cell membrane. The membrane is supposed to be like oil on water. It's supposed to be that fluid and moving. So the good cholesterol comes in here and helps create this loose thing. Okay, I always think like cholesterol is a fat, and I think butter is not the example because butter would be an LDL cholesterol, bad cholesterol. But it kind of like acts like that as far as it greases up the membrane to allow it to move better. Okay. All right. These guys, the big boxes, these are transport proteins. These are a protein. Okay. And we're going to learn of the different types of proteins. Some go all the way through. And what this does is it allows molecules to move very quickly through and bypass, and you can go in or out, bypass the cell membrane. So each transport protein is specific to a molecule that it's attached to, and it's going to be able to slide in and out of the cell faster. Some of them only exist on the inner parts. They only exist in the hydrophobic regions, and they're stuck in the middle. And then some proteins only exist on the top. So in our notes, we're going to go through all those. Okay. All right. So I also recommend that you look at the textbook. Let me grab my textbook here. Textbook does a great job, and I also tell you to go online and look at the online resources. The ebook has great models and videos to go through this, along with the homework. The homework does a great job, and especially showing those videos for you to be able to look at and see a little bit better of what's going on inside of this membrane. So make sure you're using your resources wisely. All right, I hope you can kind of see on this page. This is page 127 in your book. I got kind of a weird glare here, so I'll put my hand up. Okay. So you can see these little white things are the phospholipids. Are the phosphate, the heads. The yellow are the lipids. You see the cholesterol in the middle. You see the carbohydrate chains poking out. All right, and then these are those proteins that are stuck throughout the membrane. Some are inside, some hang outside, and then some are on top. Okay, and we're talking about the outside of the cell and then the inside of the cell. And the other thing is this fibers in here, that's part of the cytoskeleton. So remember, that's made out of those microtubules and microfilaments. So new science is showing that the cytoskeleton is attached to pieces of the cell membrane. So it helps give it a little bit more structure in here. So that's kind of a little bit more interesting stuff, that there's still more things to discover. Okay, now I'm going into the notes. Okay, for the notes. I listened to the kids in class. A lot of kids were complaining about trying to understand everything better. So I made these notes this weekend. I started on them on Friday, and I've been working on them. Probably I spent 6 hours typing up the notes to go with the chapter. I learned the chapter really well by doing this. I still recommend you do it, and it's a good way for you to learn. I'm going to teach going through these notes, but reading a textbook and making your own notes is still really important. So hopefully these notes help guide you through what's important, but still you have to do the work yourself. This type of teaching is very much what they do in college. So I know other AP teachers are teaching this way, and that's great. It definitely gets you used to college, and you know what to expect from it. Lecturing is basically how most college teachers are going to do it. I usually try to avoid this kind of teaching because I like more hands-on, and I like you guys to break up the monotony. But like I said, this is what most of the kids in class were seeming to want to go through. So I'm going to try to ever evolve into being a better teacher. So let's go through this. All right. So again, make sure you go online and you're looking at the videos. And the concepts that the book teaches. Okay. Those online videos are great. All right. The cell membrane is made out of lipids and proteins. The most abundant lipid is the phospholipid. So you are just a bunch of fat, which I find funny. All right, this word, amphipathic, is a hard word for me to say. I have to think about it. Amphipathic means that it has the hydrophilic and the hydrophobic regions. So our cell membrane is amphipathic. All right, we're also going to see that most of our proteins are amphipathic. So they also have water-fearing and water-loving regions. The stability of the cell membrane comes from sheltering those tails between the phospholipids. Like I said, that blocks things from coming in and out. Most of the proteins reside within the phospholipid bilayer with that hydrophilic region protruding. They're not random. They are in specialized patches. And those patches, even the lipids around them are specific. So we think about like the lipids that surround these big transport proteins. These aren't random, and neither are these. They're doing something that we're unclear about, which I think is pretty interesting. There's still so much we don't know about. All right, they call it mosaic, the fluid mosaic model, because mosaic means tiny little pieces. They often refer to it as like a tile. Have you ever seen a tile design where they like make a flower, they make like a pretty picture out of tiny little pieces of tile? That's a mosaic. We call it a fluid mosaic because it's always moving. The fluidity of the membranes, they're held together by those hydrophobic interactions, which is a weaker bond than covalent bonds. That's why they're able to move through there. Membranes move. Lipids can move horizontally, like somebody going through a crowded room. They can move through there. Some phospholipids even will flip over. Proteins are much harder to travel because they're really big. Some are immobile because they're attached to the cytoskeleton. Some movement is driven by the cytoskeleton, meaning they're putting it there in a particular region. And then some simply just drift. Okay, the lipid, if you think about a fat, fats have different relationships with temperature. So they're temperature dependent. If I cook beef, which I don't like to cook beef, but I will. If I cook beef, there's a lot of fat on the side. If it's hot, it's liquid. And if it's cooler or room temperature, it's solid. So your fat in your phospholipid bilayer is similar in that reaction. It depends on the species of what temperature it can handle, but you don't need your lipids in your phospholipid bilayer to become solid. You would die. It would stop movement from moving in and out. So they have to remain liquid at specific temperatures. So as temperature decreases, the membrane remains fluid if it's rich in unsaturated fat. So again, the good fat that we have helps it create that looseness, even though it might not be in the right temperature. Steroids and cholesterol will wedge in the phospholipid bilayer of animal cells. So this gives the animal cell an advantage. This allows the animal cell to go up to that 98.6 degrees, which is our body temperature, 37 degrees Celsius, and it keeps it from firming up. The cholesterol makes the phospholipid stay loose and lowers the temp, that's a typo, I'm sorry, required for the membrane to solidify. Cholesterol acts as a buffer to resist change due to temperature. So animals have that fat. Plants don't. You don't get a lot of fat from eating plants. So plants have a very low amount of cholesterol, so they're more affected by temperature change. That's why, like when we have a frost outside and the temperature, and we have a frost, that snap happens and the plant dies. It can't keep up with that temperature. The phospholipid bilayer solidifies and the plant dies. That's why our plants are changing right now as we get closer to fall. The 22nd was yesterday was the first day of fall. All right, permeability is key. They must work together to be fluid. All right, usually the membrane is as fluid as olive oil. There it is. When the membrane solidifies, permeability is changed. Enzymes become inactive, which remember, enzymes are what is going to be doing our work in our body. So we need them to stay active. Too much fluid isn't good either. With extreme temperatures pose challenges in life. And this was cool because we're going to talk about evolution. Evolutionary differences in the membrane lipid composition is basically what they've said. So we can look at the lipids of some plants and animals and say that they were designed for this temperature. This is what they evolved from. So if we look at not a fossil, but a preserved species, like if it was in amber or if it fell in a tar pit, we could look at the lipid composition and tell basically what biome it was supposed to have lived in, what it was meant for, designed for. So we see evolutionary differences in the membrane composition. Fish that live in extreme environments have high portions of unsaturated fats and hydrocarbon tails. So that's why I always said, like, fish has a good fat inside of it. This allows the membrane to remain fluid at low temperatures. Some bacteria and archaea that live in high temperatures also are going to have a different, they might have unusual lipids. That was interesting. They didn't name what it was, but they have unusual lipids. And again, this is because they've evolved to live in those environments. Winter wheat can tolerate cold temperatures because of a percent of unsaturated lipids increase in autumn. So if your wheat is harvested in the fall, it's going to have more unsaturated fat, which is the good fat. If it's harvested in the spring, it would have a low percentage. So this is a physical change due to an abiotic stimulus, that temperature. This keeps the membrane from solidifying in the winter. All right, membrane proteins and their functions. There are more than 50 different kinds of protein in a red blood cell, and the membrane is made out of mostly phospholipids, but protein determine the membrane's function. Different types of cells contain different proteins. That's supposed to be proteins, it's a typo. So integral proteins penetrate the hydrophobic interior of the lipid bilayer. So these are the ones that go all the way through the membrane. Transmembranes span, integral only penetrates the interior. So that's the one that's in the middle. Okay, so if we drew, do my drawing. There it is. So these would be the integral ones. They're only in the middle. Okay. The transmembrane protein spans the membrane. Some only extend partway into the hydrophobic interior, and they're going to fold up. They're going to collapse and coil around in an alpha helix. So there's a picture in the book that goes through what this looks like right here. Okay. You see it kind of coiling up around the alpha helix. So it's just a different shape to it. Okay. And they're made out of amino acids, 20 to 30 different kinds of amino acids. They have one or more hydrophilic channels to allow passage through the membrane, and hydrophilic substances, even water, can be able to move through here. Peripheral proteins are not embedded. They're the ones that are on top. They're loosely bound to the surface. So that's one of the ones we drew. Protein placement. Cytoplasmic side of the membrane, some proteins are held together by the cytoskeleton. The extracellular side has certain proteins that may attach to the materials outside the cell. Animal cells, membranes, proteins may attach to the fibers of the extracellular matrix. So that's between the cells. And attachments give animals a stronger framework so they can communicate faster. Signals, cells may have different membrane proteins to carry out various functions. And these are the functions of the protein. Okay, so to transport, move molecules through the plasma membrane easier. Enzymatic activity. Remember, enzymes, they speed up reactions by lowering the activation energy needed. They signal message and transduction, so they're signaling cell-to-cell recognition. That's what that carbohydrate is part of. So the proteins can recognize, like, you are an invader, we need to kill you, you're a bacteria. Or, you're when an embryo is being created, the cells will signal, like, you need to go be the heart and you need to go be the brain. And they start separating out. So they're talking to each other. And then intracellular joining. Again, cells can join up with each other to message each other and talk to each other. Attachment to the cytoskeleton and the extracellular matrix. They label it the ECM. In the medical field, there was a whole big section on HIV. So remember, HIV is a virus, and viruses have to enter a cell in order to make more of the virus. So in your cells, most people have a protein CD4. And if you have CD4, the virus attaches to CD4 and comes in through that protein. Some people have an unusual code, and they have a CCR5 protein instead of the CD4. The HIV, it goes into a little bit more complicated, like it talks about the CCR5 co-receptor, but here's basically what it is. People with the CD4 can get AIDS and get HIV, which turns into AIDS. So if you don't have this, you're basically immune to HIV. What they did is they looked at a study of people who were at high risk. They looked at people who were in drug rehab facilities, because typically people who are addicted to drugs also show a higher risk of promiscuous and risky behavior. So they have multiple sexual partners, they're sharing needles, and they're more at risk to develop HIV. So what they did is they looked at people who were at high risk. They had multiple partners and they were doing drugs, IV drugs, and they weren't testing positive for HIV, which the healthcare people would have expected them to test positive for HIV. And upon looking at this group, they discovered a new gene or a mutation that was allowing them to be successful. So what scientists are looking at is, can we manipulate this gene and do like a gene therapy on people who already have HIV? And can we basically reverse the cell's ability, like change the protein in the membrane so that new cells can't get HIV? There's a lot of cool studies that go with this. But that idea is what we're starting to look for gene therapy. All right, the role of membrane carbohydrates in cell-to-cell recognition. Remember I said this is like the antenna that hangs outside. Cells recognize each other. Animal embryo cells are going to sort into tissues and organs. So like I said, they decide who's going to become what. Basic for rejection of foreign cells, that's your immune system. So being able to say you're a bacteria, you don't come in here. And cells recognize other cells by binding molecules, by binding carbohydrates in the extracellular surface. Membrane carbohydrates. So remember that carbs are sugars. They're short. They have fewer than 15 sugars. They're covalently bonded to the lipid. If a carbohydrate and lipid are together, they call it a glycolipid. If a carbohydrate and a protein are together, we call it a glycoprotein. It varies by species, and this was interesting about the human blood. So blood types A, B, AB, and O, which we're getting ready to have a blood drive pretty soon, so that's kind of interesting to think about. If you don't know what type you are, go ahead and volunteer and donate blood, and they'll tell you what your blood type is. But you have different glycoproteins on the surface of your cell. So if you have A blood, you have A type of glycoproteins, which is what they're doing when they're typing you. They're looking at the types of glycoprotein that you have in your blood to tell what type of blood you have. And that's also why you have to bind with someone's blood type you already have. I'm type A. Remember, O is the universal donor. Everybody can take O, but otherwise I have to take A. If I get B blood in me, the B blood will come in and my body doesn't have those glycoproteins and can't recognize it and will basically see it as a foreign invader and try to kill it. And it won't do me any good. And of course, if I'm in a crisis, then it could kill me. So figure out what blood type you are. Okay, membranes have distinct insides and outsides. They differ on lipid composition, protein orientation, and they're asymmetrical, which means the inside does not match the outside. They're not mirror images to each other. All right, and that is all 7.1. All right, 7.2 talks about selective permeability, and it allows some substances to cross in and out more easily. This regulates transport across the membrane. So sugars, amino acids, nutrients, oxygen, carbon dioxide, sodium, potassium, calcium ion, and chlorine ions are going to need to get through here. The permeability of the lipid bilayer, nonpolar molecules, okay, nonpolar, polar is like water, nonpolar molecules like hydrocarbon, oxygen, carbon dioxide, and lipids are hydrophobic. So nonpolar are hydrophobic. They don't like to mix with water. They can easily cross the membrane because the tails don't like to mix with water either, so they can go through those tails pretty easy, okay? Polar molecules such as glucose and sugars pass slowly through because water can't pass rapidly because of those tails. Charged atoms also have a difficult time crossing. So things like salt are going to have a harder time, or your potassium is going to have a harder time. The lipid bilayer is only one part of the gatekeeper system. Proteins built into the membrane play key roles in regulating transport. Transport proteins, remember these are the big guys in the membrane. Transport proteins have hydrophilic substances.
We are going to resume the pledge in a moment of silence in the morning about this time. So at this time, please stand for the pledge to the flag.
Hit pause. To the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Let's pause for a moment of silence. Thank you and have a great day.
Okay. All right, so the transport proteins, they're going to be able to work on those ions. Remember, ions have a charge, and the polar molecules, and they avoid contacting the lipid bilayer by using transport proteins. We're going to use channel proteins, which look like a tube that goes through, so they don't have to touch anything. Aquaporins. The book talks a lot about aquaporins. Aquaporins facilitate water. You see the aqua there. Facilitate water. So these are really important because this is how most of our water gets through the membrane. If it had to just go through the phospholipid bilayer, it would take a long time because it's slower. The aquaporins are able to transport the water faster. Carrier proteins hold on to molecules and change shape in ways to shuttle them across. So these guys, in the video does a good job of showing it. So they open like this to take a molecule in, and they change shape to let the molecule out. Each transport protein is specific to certain molecules, and they speed up the transport across the membrane. All right, the last sections of this chapter are talking about transport in and out of the cell. We did a lot of this in honors biology. We talked about passive and active transport. We broke down the types of passive transport and the types of active transport. The good news is next week, we're doing a diffusion osmosis lab, and we get to go through all of this again. So that's going to be really good to go back through this. All right, remember with passive transport, this is again movement in and out of the cell, but this goes from what I would call a high concentration to a low concentration. And with this, it doesn't require any energy. It's easy to move from where there's a lot of something to where there's a little of something. Usually I give like a spray can or something like that. This is a can of air. The contents in here are squished in here really, really tight. They're tightly packed in here, molecules of carbon dioxide. And just by pushing the button, I can let it come out faster. There's no energy, there's no battery here. Molecules want to go from areas of high concentration out to areas of low concentration. So the molecules want to exit out because they're tightly packed in here. Same thing for your body. I just drank some water. So water. That's going to go into my stomach, and it's going to go from a high concentration to a low, so it'll diffuse throughout my whole body. No energy required to make that water from my stomach go throughout my whole body by diffusion. Okay, so it's a type of thermal energy because it's energy of motion. Diffusion is the movement of molecules as they spread out to available spaces, and it goes with the concentration gradient from high to low. Dynamic equilibrium is a balance, and here's the rule. In absence of any other forces, substances will diffuse from high to low concentration naturally. No energy is required. O2 diffuses through the cell membrane. It is continuously used for cell respiration, and so it is a continuous movement of oxygen into the cells. Water can diffuse more rapidly through aquaporins than just the cell membrane. Okay. The effects of osmosis and water. Osmosis, remember we said osmosis agua, it's the movement of water from an area of high concentration to an area of low. And this is the other way we have to think about it. We have to think about it as an area of high solute concentration. Low solute to high solute. The book gives, and in your reading, you'll see this too. These models are often used, okay? Like this, the U-shaped models. So what it's saying is, you can think of the water, but if you put a solute, and a solute is a particle, so salt or sugar over here. Even though there's a lot of water, there's more of the particle. So pure water, there's more pure water here, and there's low pure water here. So water will move from low to high. Sorry, from high to low. Sorry, high to low. So if there's pure water high here, pure water low here, water will move from the high to the low. Another way to think about it is by the solute. Here's a high concentration of solute, and here's a low concentration of solute. Water will move towards the high concentration of solute. So in this U-shaped tube, the water will move this way. So it'll actually be lower on this side, and this is supposed to be a membrane right here, permeable membrane. Water will move so that it is balanced, so that the solute concentration is the same. That's when it reaches dynamic equilibrium. So these U-shaped models are often used to try to show where the water will move. So an easier way would say you move towards the high solute. Okay. Okay. So I'm going to click down here. Water balance of cells without the cell wall. So the cell wall we'll talk about down here. Tonicity is the ability of the surrounding solution to cause the cell to gain or lose water. This depends on the concentration of solutes. They cannot cross the membrane. Let's see if I can turn this down if that helps. Okay. The high concentration of solutes, water will leave the cell. Isotonic solutions. So next week, we're going to be putting stuff in solutions, and you're going to figure out, is it isotonic? No net movement. Hypertonic will cause the cell to lose water. It'll shrivel, so it'll shrink. If increased salinity of a lake will kill animals and plants because of this. Hypotonic will cause it to swell, and the cells can burst during this. For plant cells, they have a cell wall, and that does add protection. So basically, when that vacuole swells up and gets really, really big, the cell wall starts to push back and say, whoa, 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 you're getting really close. So it pushes back and it squeezes the water out of the cell so that it doesn't burst as easily. Seawater on isotonic is isotonic. So many marine invertebrates are going to be able to use that. Remember, no net movement. Cells in most terrestrial animals, terrestrial means land animals, have a extracellular fluid around them that's isotonic, so it protects. Our protists are going to have to do osmoregulation, which means they regulate the water. Paramecium have this cool organelle called the contractile vacuole. And what that does is it squirts the water out. So it's regulating. So when the cell fills up, it starts to pump it back out. Water balance of cells with cell walls. Prokaryotes and protists, plants and fungi are surrounded by the cell wall. Rainwater is hypotonic, so it would cause it to swell. So the plants use turgor pressure, which is what I said is a way to push back, pushes back to oppose the further uptake. Turgid means that the cell's very firm. This is a healthy state for plants, and plants depend on this to be able to stand up. Flaccid means limp. So if you put, results in an isotonic. So they like the hypotonic solution because it creates that turgor pressure. If you don't have it in hypotonic, you result in kind of a limp plant. The cell wall is of no benefit of a hypertonic. It can't keep the water from leaving. The cell will shrink and can die. Plasmolysis, as a cell shrinks, the membrane pulls away from the cell wall. The plant will wilt, it can die, and bacteria and fungi also plasmolyze in hypertonic solutions. Okay, facilitated diffusion. So facilitate means to help. So this is another form of passive transport, which means I'm going from high to low concentration, going with the concentration gradient. Facilitated diffusion is going to use a transport protein to help move something in. It's very specific substances, and there's two parts, a channel protein and a carrier protein. Channel proteins are going to help move ions because they have a charge. Carrier proteins are going to change shape. So that's what I said about the molecules that change shape like that as a carrier protein. All right, in the book, page, I don't know what happened. Okay, here we go. Page 136 has a good science skills exercise to go through, which I recommend you looking at and doing. Let me know if you have any questions. Okay, 7.4. Now we're talking about active transport. So now we're going against the concentration gradient. So this is going from a low concentration of water to a high. So this is going to take energy to go against the concentration gradient. What is our form of energy? ATP. Okay, everything's going to need energy in order to get this to be done. All right, active transport uses energy to pump, and anytime you see the word pump, you should say this is going to use energy. A higher concentration of potassium ions and a lower concentration of sodium is the sodium-potassium pump. So this is going to exchange sodium ions for potassium across the cell membrane. It pumps 3 sodium out and 2 potassiums in, so they're not equal. And this takes energy to pump these guys in and out. Cells have a voltage across the cell membrane. We have an electric potential. There's a cytoplasmic side is typically a negative charge. The extracellular side is positive. So if we're moving towards a negative, then that doesn't take so much energy. But if we're moving a positive into the negative, that's going to need energy. This is caused by unequal contributions of anions and cations. The membrane potential is 50 to 200 millivolts. All right, the minus sign indicates that the inside of the cell is negative, like we said earlier. So it favors the cations, the positives, to come in because they're more attracted to each other. I think I might have said that backwards up here. Okay, so let's go through that one again. The negative side will pull positive because they're attracted to each other. So the negative inside will pull positive naturally. If we're doing negative to negative, that's going to be more of a resistance. The two forces that act on ions, we have a diffusion of ions as a chemical force and an electrical force. So we call this together an electrochemical gradient. So the electrical force is inside is negative, we pull the positives, but also the chemical force due to that concentration gradient, that high to low or low to high, because we're doing active transport. Because the sodium ions pump 2 potassiums in, a net transfer of 1 positive charge out, this process is going to store energy as voltage. The electrogenic pump, a transport protein that generates voltage across the cell membrane. In animal cells, the electrogenic pump is used. In plant, fungi, and bacteria, we use a proton pump. It actively transports hydrogen ions out of the cell. By generating a voltage across the cell membranes, it helps store energy and can help, that can be tapped for cell work. So I kind of think about like two things. One, we use the term electrolytes a lot, which is in like Gatorades, which helps bring in ions so they can move through our body that have a charge. And this is part of that, transporting those ions around and having that charge, that positive and negative. Also, if someone dies, we shock their body and send a pulse of electrical charges through the body that helps waken everything up and gets everything back to going. So our body does have electricity inside of it, and we do generate energy. Okay. The co-transplant is a transport protein, a contra supporter, and it can couple downhill diffusion with the folate of uphill transport. So basically it couples the downhill diffusion, the normal diffusion, with an uptake. Sucrose goes against the concentration gradient. That's an active transport. It couples with hydrogen ions that goes with the concentration gradient. So by coupling this together, they work bringing both parties together. The hydrogen ion was made in the ATP in a protein pump. The vascular tissue of the plant can contribute sugars to the roots and other areas. In animal cells, transport of sodium in the intestinal cells with glucose. So this is sodium with glucose, which is already moving down the concentration gradient. Sodium is pumped, so this is active, when potassium is brought in by the sodium-potassium pump. A discovery of a new pump, a sodium-glucose relationship, okay, co-transporter, I think I said that word wrong earlier, co-transporter, has helped treat diarrhea. So these two go together in this new pump, this sodium and glucose in a co-transporter. In third world countries, people still die of diarrhea, especially babies die of diarrhea. Normally, sodium is reabsorbed in the colon, which is the end of the digestive tract. But if you have diarrhea, everything leaves before the sodium can get reabsorbed. Diarrhea expels waste so fast before reabsorption. Patients are given a solute of high concentrations of salt and glucose. So in third world countries, we give patients a solution of salt and glucose. So I thought this was interesting. So what is this? Well, here is salt, sodium, and then glucose is your sugar. Here's your sugar. So the main ingredients to this bottle is salt, there's potassium, and sugar. And what is this? This is Gatorade. So in third world countries, babies end up surviving because they get our sports drink of Gatorade. I thought that was interesting. Sodium and glucose, because of that pump, that one protein that can take this in, and the relationship that they have, putting sodium in with the glucose allows it to get absorbed to your body faster. So that's what it's doing, and putting and hydrating you that way. This has lowered infant mortality worldwide. Okay, the last parts. 7.5. We're talking about bulky guys, big things, and how they are able to get in and out. They're going to use exocytosis. Remember, X is out. CYT means cell, and osis means a condition. So it's a condition of exiting things out of a cell. Endocytosis, EN is taking it in, the cell, and it's a condition of taking things in the cell. Large molecules like proteins and polysaccharides don't move by diffusion or transport proteins. They're too big. So exocytosis, the cell secretes a molecule by the fusion of a vesicle with the plasma membrane. So this is kicking something out of the cell in which it becomes part of the cell membrane. Specific proteins in both membranes are fused together to separate the bilayers. Contents spill out of a vesicle. So remember, a vesicle is any kind of membrane-bound structure with something on the inside. So when it goes from the nucleus, sends the code to the ER to build a protein. The ER ribosomes on the ER build the protein, but the ER packages and sends it to the Golgi. The Golgi takes the protein and packages it again and sends it out of the cell. This is where that protein being packaged is a vesicle. And it comes to the cell wall or cell membrane, and it fuses in the cell membrane and becomes a part of the cell membrane. All right. Pancreas also makes insulin this way. So when sugar comes into the body, the pancreas knows it needs to make more insulin, and this is going to be done the same way. It takes a lot of energy to do this. This is active transport still, using energy. Endocytosis is taking a big molecule into the cell. All right. And then there's 3 types of endocytosis, and I've got some videos for you to watch these. Phagocytosis, P-H-A-G-O, means to take it in, to ingest, phagocytize it. Cyte is cell, and osis is a condition of taking food in the cell. Pene means to pinch or push out. So penocytosis is kicking it out of the cell, like drinking water. And receptor mediated endocytosis. So humans use this to take in cholesterol and make steroids. Cholesterol travels in the blood as an LDL. An LDL cholesterol is a bad cholesterol. We want low numbers of LDL. LDL rebinds to receptors in plasma membranes. Hypercholesterolemia, high blood pressure, is an inherited disease, gives high levels of cholesterol in the blood. LDL receptors are missing. So this is the bad cholesterol that we don't like. And if we don't have the receptors to bind to take this out of our blood, then we get strokes and heart attacks. So it sticks to those arteries and stays there. Endo and exocytosis also help in rejuvenating the plasma membrane because it's adding the membrane back into the cell. Okay. The book has some good pictures here. Okay. All right. So here, this is phago. We're eating food. So the cell membrane is surrounding and pinching and making a vesicle and is coming in. And it doesn't show, there's exocytosis, sorry. Exocytosis, that vesicle is moving to the piece of the membrane and it opens up and spills it out. And then this membrane becomes part of the cell membrane. So that's how it keeps like making it fresh and going back and forth. Okay, so you need to read chapter 7. About half of you admitted that you read and spend hours on the book, which is good, and the other half are not even touching the book. So make sure that you are touching the book. Read over it, especially after watching these notes. You have a homework and a practice test on the Pearson website. Make sure you're completing those. These homework questions are great. They have videos. They have areas for you to go back and read, and it re-emphasizes the book. Read the book first, do these notes first, and then do the homework. Then take your test. I will count this test. Complete the Unit 2 progress check on the AP Classroom. I need to know how you're doing on AP questions. And that's all you have to do this week. All right, guys. I'm going to somehow exit all of this out. And I hope you have a great day and a great week.